Hello, welcome back to the third module. So, what I have been discussing about the importance of beta carotene and what needs to be done to produce more beta carotene in plants using genetic engineering tools. Let us go to the slide. So, this is basically showing the countries where populations are showing vitamin A deficiency and which is the cause of the night blindness. So, here you see here that a baby is given a drop of vitamin A so as to alleviate the vitamin A deficiency, and this contains this really is responsible for the malnutrition, or in other words that malnutrition is responsible for this. So, now how to cope up with this? Before that you see the severity of this, that vitamin A deficiency leads to xerophthalmia, which is the night blindness. Now, it is happening particularly if we concentrate our discussion on the South East Asian countries. This is very prevalent. Now, why? Because the economic conditions are not that good as compared to the other part. So, here people are mostly dependent on eating rice, and they may not get, and mostly they are eating rice only because they do not have sufficient economic power or to provide or to get the access of a balanced nutrient. So, in other cases they are mostly eating rice 3 times a day. Okay. So, eating rice will apparently fulfill their hunger, but their rice cannot really fulfill a balanced nutrient content, and particularly the deficiency of vitamin A will continue at such a juncture that eventually it forms xerophthalmia, particularly to the younger population. So, to prevent that you need enhanced vitamin A content, or to fulfill that vitamin A deficiency you need more beta carotene to be produced in the food, in the staple food. Okay, so that the beta carotene can be converted into vitamin A. As I told you before, that if we consume sufficient amount of beta carotene, then our body system has the enzyme which will convert this beta carotene into vitamin A. So, keeping that in mind, scientists attempted to create golden rice that will alleviate the malnutritional problem, particularly in Southeast Asia. So, in other words, the beta carotene can be incorporated, if it can be incorporated to rice, to maize, to cassava, then it will be really great. Now, let us see whether it is possible to incorporate beta carotene genes into rice. Now, the milled rice what is available, that is provitamin A free. So, there is no trace of any precursor of vitamin A. And this is what mostly people are consuming, and as a result of that we get night blindness, xerophthalmia, and other several other diseases including diarrhea, respiratory diseases, measles, soap and so on. And epidemiology is quite severe. So, what has to be done? So, if we can make the rice nutritionally rich by adding beta carotene component into it, then that will be alleviate the nutritional problem. So, the figure showing here is basically the plastid chloroplast where the pathway is operating, and this is a detailed pathway of the beta carotene biosynthesis, which I have discussed partly in the previous module when I talked about the gene expression in tomato, leading to more nutritious tomato. Now, whatever we desire to achieve, it is not always possible to achieve, because plants do not underestimate plants, that whatever we would like to do, plants will be able to do. Plant also having certain limitation. So, what is the limitation in the rice plant with this pathway? The limitation is this: the rice plant, in rice plant, the pathway beyond geranyl diphosphate is non-existent. In other words, there is no existence of phytoin synthase, phytoin desaturase, zeta carotene desaturase and lycopene cyclase. The whole pathway is missing in the rice. So, if the whole pathway is missing, how it is possible to transfer the genes of beta carotene into rice? In case of tomato, what we have seen, that okay, we putting one gene, it function, but here we have to put in multiple genes, and that too we are not sure whether this gene will function or not. So, the only option is to transfer all these genes into rice, now which is not very easy job. So, phytoin synthase, we have to get the genes. So, where from we will get the genes? So, we have to find out the source. You all know the daffodil, Narcissus pseudonarcissus, that is the scientific name. Daffodil plants produces carotenes in its flower, and this phytoin synthase gene has been cloned and characterized by a scientist called Peter Baer at the University of Freiburg in Germany. Similarly, the lycopene beta cyclase genes are also available with Professor Peter Baer, and phytoin desaturase and zeta carotene desaturase are also there, but instead of taking these two genes based on previous experience, why not to think of hiring the Erwinia, that is the bacterial CRTL genes, which if it express can convert phytoin into lycopene by a single step, as it was tested for tomato. So, in other words, we need phytoin desaturase, we need CRTL, which is converting phytoin to lycopene. We also need lycopene cyclase from daffodil, and these all genes to be incorporated into rice. Now, Peter Baer is a molecular biologist, he has all the gene sources, but to transferring genes is in rice is not an easy job, multiple gene transfer, moreover it is monocot. So, an ambitious project was created, was undertaken by Ingo Potraikas of, he was the group leader at Swiss Federal Institute of Technology in Zurich, which is called ETH Zurich. So, Ingo Potraikas is a man of plant transformation. So, he standardized the rice transformation technique using agrobacterium. So, both Peter Baer and Ingo Potraikas joined hand, and they took that ambitious goal to create golden rice or to transfer the beta carotene genes in golden rice. In rice plants, sorry. So, now the point is this, to transfer these genes into plants. So, as I said to you before, that we need a phytoin synthase, that source will come from daffodil. We need a CRTL, that will come from Erwinia bacteria. So, they made two different constructs. In one, they attempted to make this rice accumulating lycopene. If rice accumulating lycopene is red in color, like the tomato, traditional tomato color. So, why not to make rice accumulating lycopene? Because if you create rice accumulating lycopene, so that will be very nutritionally rich and it is a very strong antioxidant. If that can be fed to the people, that will also be good. So, that they have made a single construct between the border sequence. So, they put a APH4, which gene is a basically a hygromycin resistance gene, which act as a selectable marker. They put phytoin synthase gene under the control of a glutenin promoter. So, this promoter will allow to express this gene in the rice endosperm, and they put the CRTL gene under control of a constitutive promoter. So, throughout the plant it will express. They also put an additional antibiotic resistance gene, which is neopyrazine phosphotransferase, which can give which will give resistance against kanamycin. So, this entire construct was put it into binary vector in Agrobacterium, as I told you before, and then how it has been transferred. So, the immature rice embryos were taken, and these embryos were inoculated with the Agrobacterium suspension containing this construct, and then eventually it will put into the tissue culture medium, and then transform plants were grown. They were selected on the basis of hygromycin, and then they check for the presence of this gene. They studied the expression, and then they look for the presence of lycopene. What is the aim of this? Now, if we concentrate here on this one, this is what I have written is called co-transformation. So, here two constructs were created. In one construct, they put phytoin synthase gene under the control of glutenin promoter and the CRTL under the control of a 35S constitutive promoter, and the other construct they made where they put the antibiotic resistance gene, hygromycin resistance, and the lycopene beta cyclase. Now, these two constructs they put in two separate binary vector and in two agrobacterium, two different agrobacterium. So, these agrobacterium were grown as a suspension, these agrobacterium containing this construct was grown as a suspension. So, these two agrobacterium were grown, and then once they grown at the log phase, they were mixed together and it was used to do transformation utilizing immature embryo of rice. So, that is why it is called co-transformation. So, when the transformation was carried out, it was carried out with simultaneous presence of both the agrobacterium. This is what is shown in colored diagram. So, this was the original construct diagram published by Potraikas and Peter Beer in the journal Science in 2000. So, the same thing I have discussed before. So, this is for the self study, you can look more details into the construct. So, the color code remains same, so that it will be easy to follow. Now, before we come to these results, for single transformation it is just straightforward, but the co-transformation it can happen that the tDNA from one agrobacterium go and integrate, whereas the other one may not integrate. So, how to select rice plant containing genes from both the agrobacterium? So, they did it first on the basis of antibiotic resistance. If I go back to here, so first they check for the transform plant whether they are surviving in presence of hygromycin or not. So, those which are not surviving, they simply discarded. So, these population the populations which are surviving in presence of hygromycin. So, these contains maybe the rice plants which have both the tDNA inserted from both the agrobacterium or may contain only one. So, next what they did, they check carefully the presence of these genes into the hygromycin resistant plants, and they found several plants where these genes are not present, they simply discarded. They found only a few population where these genes are present. So, the population where these genes are present, so this gene was also present. So, those plants were taken and those plants were called co-transformed plants where all these 3 genes were present. Okay. Now, what they did the analysis. So, they grow the plants, the plants were allowed to grow and then they flowered and eventually they formed the seeds. So, the seeds they harvested and then what they did, they made the removed the husk, they polished the grain and then the grain looks colored, and then what they did the extraction with solvent, and using high performance liquid chromatography method they analyze the carotene content. First they analyze the carotene content in the single transformants, where there was, to remind you, there was no gene for lycopene cyclase. So, there was only gene for lycopene formation, that is the genes for PSY or phytoene synthase from daffodil, and CRTL, that is genes from Erwinia, which will zeta carotene desaturase, which converts phytoene to lycopene. So, they were looking for the presence of lycopene, but what they found, to their surprise, that lycopene is there, but beta carotene is also there and in sufficient amount. That is also very interesting, because they did not put the genes for beta carotene, they expected only lycopene to happen, but they found beta carotene also. That means what happened? That definitely lycopene was produced and that perhaps induced the lycopene cyclase activity leads to beta carotene. Exactly what has happened in case of tomato, what I discussed in the earlier module. Now, when they looked into the co-transformed plants, here also they collected the seeds, okay, and seeds, the husk were removed and it was polished, and then they did the analysis and they find that of course, it is beta carotene is present and other carotenes are present. So, the variation is much more here when compared to the single one. It is obvious because here you have all the 3 genes, phytoene synthase, zeta carotene desaturase as well as lycopene cyclase, and in the control there is no carotene formation at all. So, these were all possible by utilizing the tissue culture techniques. So, without tissue culture techniques you cannot make successful transformation. So, the take home message is that if you want to do any transformation work, you should have a regeneration system ready, and only those plants you can attempt to do agrobacterium transformation. So, pathway complementation. So, what is this is the co-transformation. What you see that one of the construct only contains lycopene cyclase and the other construct contains phytoene synthase and CRTL, that is zeta carotene desaturase. Okay. So, with the selectable marker genes only into all yellow transgenic seeds expressed lycopene beta cyclase, and they all contain beta carotene, which was expected outcome of course. But there was a second construct which was a simple construct without lycopene cyclase. So, there also there is no lycopene cyclase, but it produced golden rice instead of red. Red means that it should contain more of lycopene, but it produced golden rice. That means, without inserting lycopene cyclase, it produces golden rice yellow color. So, that is surprising, but a scientific fact. So, now if you see the golden rice created, we call prototype, because this is the first one created as a result of collaboration between Peter Bayer and Ingo Potraikas. So, the control seeds are shown in white, and these are the different populations like these are the with single transformants and this is with the double transformants co-transform lines. So, the here intensity is much more because you have the genes for the beta cyclase. This is a real picture and these pictures are there in the Science journal. If you download the original paper, you can get these things, you can read the original paper. So, this is there, I have taken from that source. So, this gentleman is Ingo Potraikas, okay, and it came in through the cover issue of Times. So, the title of the paper was Engineering the Provitamin A biosynthetic pathway into rice endosperm, carotenoid free rice endosperm. So, this was published in 2000 and the very novel thing they did not patented this genes or this technology, because they wanted that the output should reach to the entire population or the poor people without any hindrance, so that it can be grown freely and the outcome of the scientific discovery can benefit to the population at the ground level. Now, when this was made, then there are a lot of effort both from the public laboratory as well as the private laboratories. Now, the public laboratory they did not make patent, but then they started further work on this. They want to make more intense golden rice, and for that they started manipulating the constructs, okay, either changing the different promoters or changing the source of the genes, but basically keeping the same genes. You cannot change the gene, but you can change the source genes. Like daffodil was used for lycopene cyclase, daffodil was used for phytoene synthesis. Now, if the source gene, if the source can be changed into other plant. So, I will tell about that. So, like here it is written ZM. So, Gia maize. So, maize eventually the Gia maize phytoene synthesis was used instead of instead of daffodil. Now, before I go further, so scientists were also interested to know that the rate limiting step. So, they did lot of gene constructs, they did more with different promoters, okay, beta cyclase from a range of plant sources, but eventually they found that it is basically the phytoene synthase which makes the trick. So, phytoene synthase is the first one which convert GGPP to phytoene. As I said to you, in the rice endosperm, GGPP is produced for different purpose, but beyond GGPP nothing is produced related to beta carotene synthesis. So, the phytoene synthase is that enzyme which converts the endogenous GGPP available into phytoene, that is the first product of the beta carotene pathway. So, scientists with different trial and error, and here I must say that the private partners like Syngenta, they started now entering into this venture. So, what they propose that they will utilize their resource, expertise, money, and whatever comes out that they will utilize for commercial purpose, whereas the prototype one is already available free. So, anyway, so with their effort finally they were able to achieve golden rice 2 by using a construct where they have used Gia maize phytoene synthase instead of lycopene. And that paper was published in Nature Biotechnology 2005, improving the nutritional value of golden rice through increased provitamin A content. So, they did a lot of experiments. So, some of the experiments are listed here. So, you can read the Nature Biotechnology paper that is available in the public domain, and I will discuss the compare what is more there in the golden rice created by the Syngenta in the subsequent slides. So, these all slides are showing the enhancement of the carotene contents with this specific construct, and when you see the image here, there is a clear difference on the intensity of the color in these two. First one is the control, this is the golden rice 1, this is the golden rice 2. So, in between they created a golden rice 1, so which showed improved color intensity than the prototype, but not that much difference, but in fact, the golden rice 2 created by the private sector, the difference is intense. So, what is the difference between golden rice 1 and 2? So, the golden rice 2 can produce 35 microgram of carotenoids per gram dry weight, whereas the golden rice 1 produced 1.6 microgram of carotenoids. So, you see the color difference. So, it is huge. Okay, so, more efficient phytoen synthase gene was used, the source was Giamese, not Narcissus. So, other thing they did it is that earlier CAMV35S constitutive promoter was used, but here they have used a polyubiquitin promoter. The most important thing what they have used, that they have removed the antibiotic resistance marker gene, because there was worldwide concern of transgenic food crops containing antibiotic resistance gene. Instead, they have used a selectable marker genes, which is called a positive selectable marker phosphomannose isomerase, a sugar based system. So, the beauty is this phosphomannose isomerase gene will code for phosphomannose isomerase, which converts mannose to fructose. So, normally plants cannot grow with mannose. So, in the medium, if you add mannose and if your transgenic plant contains phosphomannose isomerase genes and if it expressed, then it encodes the phosphomannose isomerase active enzyme, which will be able to uptake mannose and convert it into fructose, and that fructose can be straight away utilized for the plant growth. So, it is called positive marker. So, by that way antibiotic resistance genes can be removed. So, human trial has also been made, and it basically showed the consistency as recommended by the dietary allowance requirement for beta carotene. So, to sum it up, beta carotene pathway problem, whatever existed in rice, has now been taken care of. The genes from different sources, for example, daffodil or Giamese, Erwinia, daffodil, whatever required, were successfully inserted in rice, and eventually that leads to the production of very intense color golden rice and reaching a very high content of carotenoids that will fulfill the malnutritional requirement. But little unfortunate part is there, because this is created by the private sector. So, it is not freely available, but the government can venture into it, and with their significant input in it and assistance, this can be popularized. And in India, these trials were successfully conducted in IARI, TNAU, Directorate of Rice Research, It was conducted in Bangladesh, Philippines, Vietnam. So, now it is waiting for the clearance before it released to the market. So, there is no inherent right or wrong way to enhance nutritional content, but there are some oppositions that they said the GMO is not safe, but there are both pros and cons behind it. So, being a scientist, it is just a piece of DNA. So, and with DNA does not make it is a chemistry, it is a chemical. So, where from it comes, and eventually it is a very tiny piece, and when plant propagates, that is eventually lost. So, the fear of GMO will eventually go away, and when the public sentiment is important. So, when public, there definitely a group of public is ready to accept, and we believe that in the next decade we should see that golden rice are growing freely, and that will help to alleviate the malnutritional problem worldwide, particularly in the developing countries. So, with this I will be ending my lecture on the enhancement of nutrient contents by biotechnological tools. So, there are different tools available nowadays like genome engineering, CRISPR based. So, this lot of progress have been made, but what I have basically discussed in this course is the classic methods, which are foolproof method, well proven and scientifically tested. And because this is a refresher course, and I believe that the teachers who will be taking this course in distant mode, so they get enough confidence, so that they can convey some of these information to their students at undergraduate and postgraduate levels, and then will feel more delighted that our effort goes for positive, in a positive way, for making manpower trained with latest scientific discoveries. Thank you.